microplastics move into organisms' tissues where they can cause inflammation and oxidative stress. These effects can vary based on the routes of exposure, the particle properties and concentration, and the organisms themselves. Despite growing concerns about plastic pollution in the Great Lakes, there is not currently a coordinated regional effort to monitor and tackle microplastic pollution in the basin. This means there isn't a way to definitively say whether microplastic pollution in the Great Lakes is currently good or bad, or whether the situation is improving, worsening, or changing over time. The IJC and its expert advisory boards have been focused on Great Lakes microplastics for a decade. The latest report builds on previous work by the IJC and other organizations. The first peer-reviewed study to measure and report on microplastic pollution in the Great Lakes was published in 2011. The IJC's Great Lakes Water Quality Board have maintained a microplastics watching brief since 2015, and the Water Quality Board's watching brief summarizes information related to Great Lakes microplastics and plastics more broadly, spanning science, policy, and media. In 2016, the IJC hosted a workshop to develop recommendations to address challenges posed by microplastics in the Great Lakes. Based on that workshop, the IJC published a report in 2017. That report outlined 10 recommendations for Canadian and US governments to reduce and monitor microplastics pollution. This Science Advisory Board project aimed to produce a state of the science about the environmental effects of microplastic pollution in the Great Lakes. The project had two main goals. First, to develop standardized sampling methods for monitoring microplastics. And second, to develop risk assessment and management frameworks for microplastics in the Great Lakes that are tied to their ecological health impacts. It is important to emphasize that the scope of the project only examined the science of the environmental and ecological impacts of microplastics. Science on the human health impacts of microplastics was outside the scope of work for our project. This project focuses on the impacts to water, sediment, and biota, which includes plants, fish, wildlife, and organisms living in the Great Lakes aquatic environment. Significant work in the last decade has focused on the science of environmental and ecological impacts of microplastics in freshwater habitats. We brought together science from local and global resources. In 2018, the California legislature passed two bills that required the creation of tools to, 1, collect environmental samples for microplastics-related analysis, and 2, quantify and characterize microplastics in environmental samples, and 3rd, assess and manage risk. I'd like to bring focus specifically to the work by the Southern California Coastal Water Research Project Authority, or SQURP, as we call them affectionately. Since the bills in California were passed, SQURP has convened several workshops and work groups to develop these tools. Our work group adapted those tools for the Great Lakes, which you will hear more about shortly. I should also mention that many of the people involved with SQURP's work group were also involved in our project. I'm going to turn things over now to Dr. Chelsea Rockman, who will start our discussion of the report.
Good afternoon, everyone, and thank you, Rebecca. My name is Chelsea Rockman, and I'm a professor in ecology at the University of Toronto. There were three main objectives for this board's project. First, we wanted to synthesize recent literature and the science advances and knowledge about microplastics in the Great Lakes. We also wanted to develop a framework for monitoring microplastics to enable harmonized reporting efforts across the Great Lakes region. And finally, we wanted to develop a risk assessment and management framework to better indicate when management actions are needed. In addition to finding, in addition to the findings highlighted in the board's report, the report's supplemental material provides details on how the report was developed, our proposed standard operating procedures for sampling, tools relevant to our proposed risk assessment and management framework, as well as an extensive literature review with 2 databases. To meet our first objective on synthesizing recent scientific advances and knowledge the work group undertook, we first had a session on plastic pollution at IAGLR, or the International Association of Great Lakes Research, in 2023, which was held here in Toronto. Next, we wrote a literature review, which includes sections on the physical and chemical characteristics of microplastics, field and lab methods, environmental occurrence data for the Great Lakes, and toxicity data for freshwater species, as well as regional policy and management considerations. And finally, we added the most recent environmental and toxicity data to publicly accessible databases, and those QR links here will take you to those databases. For the data on microplastics in the Great Lakes Basin, our updated database is available at U of T's Dataverse, which is publicly accessible to anyone. And for the toxicity data for freshwater species, our work group helps update an existing database of microplastic toxicity studies called the Toxicity of Microplastics Explorer, or TOMEX, which we developed as part of this work group. TOMEX 2.0 is not currently available now, but it should be available soon in 2025. TOMEX 1.0 is available now, but 2.0 will have toxicity data from over 300 studies on both marine and freshwater species. The project reported on the presence of microplastics in water, sediment, living organisms, or biota, and on shorelines in the Great Lakes and in their connecting river waters and tributaries. So this is data in the report, the literature review of the report. So the figure here shows the concentrations of microplastic particles reported in surface water across the studies in our literature review. The concentrations per cubic meter range from less than 10 particles to approximately 10,000 particles in offshore waters and upwards of 100,000 particles in tributaries. Microplastics have also been found to contaminate algae and to occur in fish, birds, amphibians, as well as invertebrates. Generally, the most populated lake basins, like Lake Michigan and here at home in Lake Ontario, have the highest concentrations of microplastics in water. Additionally, tributaries generally have higher concentrations compared to the open water. Our project also explored the sources of microplastics and how they travel through the environment, looking at both land-based and aquatic releases. Land-based sources include tire and road wear particles, microfibers from textiles or our clothing, microbeads from personal care and cleaning products, as well as pre-production resins from industrial facilities, and mismanaged plastic waste or litter from urban and agricultural areas. Aquatic sources would include recreational activities like beach visits, fishing, boating, and other activities like commercial fishing and shipping. We also know that microplastics move into the Great Lakes via various pathways like stormwater and runoff from urban and agricultural land, from wastewater effluent, from tributaries, through the wind, and from far away via atmospheric deposition. And in our work, it became really clear that there was no globally widely accepted definition for microplastics. And this means the data in reports and studies comparing them can sometimes be a little bit like apples and oranges. And with a common definition, it's really important to reduce existing challenges for effectively monitoring, putting that data into risk assessment, and using it for management. So this report proposes a size and composition-based definition of microplastics, which is based on California's State Water Resources and Control Board definition, which again came from that SWRP working group. It has been modified to include particles only greater than 1 micron, which reflects the current focus of microplastics monitoring programs in California and what we're recommending here in the Great Lakes. So I will now hand things over to Eden Hatley, who will talk about the work's second and third objectives.
Thank you, Chelsea. Good afternoon, everyone. My name is Eden Hatley. I'm a PhD candidate in environmental science at the University of Toronto. The current approach to understanding microplastics in the Great Lakes is highly varied. While there is strong evidence to suggest that microplastics are found throughout the Great Lakes Basin, much of this evidence is based on published, peer-reviewed data from discrete projects on research initiatives. This sampling is not systematic, often geographically limited, and data are not routinely collected using harmonized methods. A regional, coordinated approach is needed to systematically monitor microplastics. A routine, basin-wide monitoring program would generate data using harmonized protocols to help identify trends in microplastics presence. This is essential for informing and evaluating the effectiveness of policies to manage and mitigate sources of microplastic pollution. This work group's second objective focused on developing a framework for sampling microplastics in different media, lake and tributary waters, sediment, and biota, living creatures like plants and fish. In September 2023, the work group hosted experts to develop recommendations for harmonized monitoring of microplastics, including 1, methods for sampling microplastics in the environment, 2, program design to track spatial and temporal variability as well as transport pathways and sources, 3, opportunities to integrate microplastics into existing regional contaminant monitoring programs. To meet our second objective, the work group has proposed standard operating procedures, or SOPs, for monitoring microplastics in lake water, tributary water, sediment, and aquatic biota. The QR code on the slide links to these SOPs. These SOPs aim to standardize monitoring across the Great Lakes and align with similar efforts in North America, including California and Chesapeake Bay. To achieve this alignment, we adopted existing SOPs developed by SQUIRP for lake water, sediment, and biota, and created Great Lakes-specific addenda for each. For tributary water, we wrote our own SOP. The work group's final objective was to develop an ecological risk assessment and management framework for microplastics in the Great Lake Basin. In January 2024, the work group convened a second expert workshop to develop recommendations for 1, an evidence-based ecological risk assessment framework to determine ecologically relevant thresholds for management action, and 2, guidance on incorporating exposure and effects data into this framework. Again, to align with similar efforts across North America, the workshop focused on adapting an existing risk-based management framework for microplastic pollution, recently developed by SQUIRP, released in 2022, and shown on this slide, to ensure its relevance to the Great Lakes. SQUIRP developed a tiered risk-based management framework for microplastic pollution. The tiers are divided by 4 risk threshold values for microplastic exposure in water, derived using data from lab-based toxicity tests. We developed an adapted risk management framework for the Great Lakes, shown here. This framework aims to contextualize monitoring data and support the potential inclusion of microplastics as a toxic chemical sub-indicator in the Canadian and US government's State of the Great Lakes reports, or SOGL reports. Under the Great Lakes Water Quality Agreement, the government's SOGL reports are published every 3 years. The report is the government's assessment of the status and trends of various chemical, physical, and biological indicators and sub-indicators of ecosystem health. The governments track and report on indicators of toxic chemicals in their SOGL reports. Under the SOGL Toxic Chemicals Indicator, ecosystem health status is assessed by comparing chemical concentrations to threshold values of concern or established guidelines. Long-term trends in ecosystem health are evaluated based on concentration changes over a 10-year period, recognizing that many of the sub-indicator chemicals are persistent in the environment. Our proposed framework includes 3 tiers, categorizing ecosystem health as good, fair, or poor, based on increasing microplastic concentrations. It also tracks ecosystem health trends over time and defines 2 thresholds values of concern. This categorization is designed to follow the assessment structure of the government's indicator assessment and reporting that they already use in their SOGL reports. To derive the 2 risk threshold values used in our framework, we applied the same risk assessment framework as SQUIRP, which is based on the species sensitivity distribution, or SSD approach. SSDs are statistical distributions that illustrate how species vary in their sensitivity to a specific substance, in this case, microplastics. Each point on the distribution represents a species' response to microplastic exposure in a lab-based toxicity test. Species at the top of the distribution are least sensitive, while those at the bottom are most sensitive. SSDs are commonly used by governments to establish safe limits for contaminants. By analyzing the distribution, we can determine the concentration at which a given percentage of species in a community, typically 95%, is expected to be protected from harmful effects. This slide shows our SSD for microplastic exposure in water. We use the TOMEX 2.0 database to construct this SSD, incorporating available data for species either found in the Great Lakes, such as the common duckweed, or species similar to those residing in the Great Lakes, such as the small freshwater fish Medaka. We identified 2 threshold values from this SSD to use in our risk management framework. These threshold values correspond to the hazard concentration values HC5 and HC30, which estimate the microplastic concentrations at which 5 and 30% of species in a community would be affected, respectively. We performed a preliminary comparison of these thresholds with microplastic concentrations reported in Great Lakes waters. This figure on the slide illustrates this comparison. This figure shows the cumulative distribution of microplastic concentrations across each Great Lake. By analyzing it, we can estimate the likelihood that a water sample will have a microplastic concentration either below or above a given value. Our preliminary comparison suggests that fewer than 20% of water samples from Lakes Michigan and Ontario will have microplastic concentrations exceeding our upper threshold, while nearly 100% of water samples from Lakes Superior, Huron, and Erie will fall below our lower threshold. It's important to note that this comparison is preliminary, as we currently lack sufficient monitoring data for each lake, and the existing data were not necessarily collected using fit-for-purpose methods. This again highlights the need for a routine, basin-wide monitoring program that generates data using harmonized protocols. At the time of the workshop, the available data were too limited to conduct a preliminary assessment of microplastics in sediment. I'm going to turn things over now to Dr. Karen Kidd to walk us through the key recommendations from this report.
Thank you, Eden. It's wonderful to be here. My name is Karen Kidd. I'm a Science Priority Committee member and a professor at McMaster University in Hamilton, Ontario. Based on our review of the state of the science, the patchwork of research that's been done on microplastics in the Great Lakes has revealed gaps that need to be filled. It's not currently possible to establish the status and trends of microplastics and their impacts on the Great Lakes ecosystem, and the issue needs systematic monitoring. And without indicators, it's not feasible to evaluate the effectiveness of efforts to clean up and prevent microplastics pollution. As the saying goes, you can't manage what you don't measure. Therefore, the board has advised the IJC of 4 recommendations that the United States and Canadian federal governments could pursue to address these gaps, and they are shown on the slide. As the previous slide showed, sampling effort is not consistent across the lakes and not yet adequate to rigorously assess ecological risk or trends over space and time. We first recommend the governments develop and implement a regionally coordinated monitoring program for microplastics, one which follows harmonized methods and adheres to consistent reporting guidelines to inform the ecological risk assessment, mitigation, and management of microplastics in the Great Lakes. The SOPs, or standard operating procedures for monitoring, and the risk assessment and management framework developed through this project were designed to be feasible to implement within the government's existing coordinated science and monitoring efforts. Second, we recommend the United States and Canadian governments support continued research to enhance monitoring and analytical capabilities. This includes studies on better laboratory methods to understand the sources and characteristics of microplastics, improve their detection, and increase sample throughput. This research should also include advancing monitoring techniques and expanding our understanding of the ecological effects of microplastics on aquatic species relevant to the Great Lakes, particularly from exposures in the sediment. Next, to help coordinate monitoring, assessment, and management actions, our third recommendation is that microplastics be evaluated under the Great Lakes Water Quality Agreement as a toxic chemical sub-indicator and as a chemical of mutual concern. And finally, our fourth recommendation is that steps should be taken to prevent the emissions of plastics and reduce plastic pollution in the Great Lakes Basin, thereby reducing environmental concentrations and preventing further ecological risk. I would like to take some time to expand on our third and fourth recommendations. Through the Great Lakes Water Quality Agreement, the United States and Canada commit to protect, restore, and enhance water quality in the Great Lakes. The agreement is a powerful tool for coordinating federal resources and basin-wide action to tackle water quality threats and safeguard the lakes for those who live and work and play in the Great Lakes region. The agreement outlines 9 objectives for the Great Lakes water quality. Objective 4 aims to ensure that the lakes are free of pollutants in quantities or concentrations that could be harmful to human health, wildlife, and aquatic organisms through direct exposure or indirect exposure through the food chain. Progress towards the agreement's objectives is measured using indicators and supporting sub-indicators. The indicator for Objective 4 is toxic chemicals in the Great Lakes ecosystem, and these chemicals are systematically monitored and reported on as part of the State of the Great Lakes report. The table you see here is from the 2022 report. It is a summary of the status of toxic chemicals within different compartments, such as whole fish and water, in each of the Great Lakes. The monitoring framework, as well as the risk assessment and management framework for microplastics that we developed, were designed specifically to dovetail into the government's existing indicator reporting process, some of which you see here. The toxic chemicals that governments currently monitor to assess progress towards Objective 4 include chemicals of mutual concern, also known as CMCs. CMCs are chemicals that originate from human sources and are potentially harmful to human health or to the environment. Currently, there are 8 contaminants designated as CMCs. For example, one CMC that you're likely familiar with is mercury. For a chemical of mutual concern, governments develop strategies for risk management that may include binational or domestic actions such as research, monitoring, surveillance, and pollution control and prevention measures. So not only do Canadian and US governments conduct systematic, regionally coordinated monitoring for each of the CMCs, but they also develop plans to prevent and clean up CMCs in the Great Lakes. The findings from our report provide compelling evidence that designating microplastics as a CMC is an important next step to understanding and taking action to prevent future harm. The governments are currently considering a nomination they received in August of 2023 for microplastics to be designated as a CMC under the Great Lakes Water Quality Agreement. It is up to the Canadian and US governments, not the IJC, to make that determination. Should governments designate microplastics as a CMC, part of that process would be for governments to outline actions aimed at reducing their release into the environment. Therefore, our third and fourth recommendations go hand in hand. As a CMC, there would effectively be a strategy in place to prevent and mitigate microplastics pollution. And as a sub-indicator in the State of the Great Lakes reporting, the systematic harmonized monitoring would provide much needed information to evaluate if these actions are effectively improving the Great Lakes environment and achieving the objective for the lakes to be free of harmful pollutants. I'm going to turn things back over to you, Matthew, now to kick off our question and answer period. Thank you.
Thank you all for your remarks and the helpful information that you've presented. We will now open the floor to questions from the audience. As a reminder, for folks participating in the Zoom webinar, please use the Q&A Zoom function to enter your question. The chat will not be available. For folks watching the live stream, please send your questions via email to the address listed on the screen. We will do our best to answer as many questions as we have time for today. but we may not get to all of them. If we run out of time and your question has not been answered, or if you have a supplemental inquiry to your original question, you can email that along and we'll certainly coordinate a response. So I've been keeping an eye on the Q&A as the presenters presented their material. We're taking a little bit of artistic license in that some of the questions are very similar to one another. So in those cases, we'll attempt to lump those questions. If you don't hear your question exactly as you wrote it, hopefully you will see the sort of spirit and intent of your question reflected in some of the questions and answers that we get into now. So one of the questions, a couple of them actually, related to translocation of microplastics to organism tissue. And there was a very specific inquiry relative to dreissenids, to zebra and quagga mussels. But I'm just wondering perhaps if, Chelsea, you might start off with the current state of science relative to translocation of microplastics to organism tissue.
Sure. So I would say that the science, I feel like now there have been enough studies that have found microplastics beyond the gut content of organisms, suggesting that they do indeed somehow through some mechanism get outside the gut into different tissues. So they've been reported in livers of fish, they've been reported in the muscle or the fillet of fish. I believe Karen has done some work looking at it in mussels. So I think the mussel specific question I'll hand over to Karen. But I think what I would say we understand right now is they do translocate, they do get outside of the gut, they can move through the body. The mechanisms we're still trying to understand. The rate of translocation we're still trying to understand. And whether or not they bioaccumulate or increase over time as the organisms age, we're also still trying to understand. Karen, do you want to riff off of that?
Yes, thank you. I'm not familiar with anyone that's working on quagga mussels or zebra mussels and microplastic uptake, but I imagine someone is. We've been doing some work in the Grand River, which flows into Lake Erie, and looking at microplastics uptake into mussels. And we do see that they accumulate in the mussel. And as Chelsea mentioned, they do translocate out of the gut of the mussels and into the body. So this is a widespread phenomenon that we're starting to get a better handle on through these kinds of research projects.
Thank you. There's a number of questions related to sources of microplastics. There was a very helpful figure included in one of the slides that spoke to this. But Eden, I'm wondering if you might elaborate a bit on sources of microplastics, including things like bottles and containers and things that are in use in everyday life, and to what degree they might transmit plastic particles into organisms, et cetera.
Sure. So it's not always feasible to determine where a microplastic came from. So often what's found in the environment is so small, such an odd shape that maybe the original material that it was part of, it's not possible to determine. But from our review of studies that have done monitoring in the Great Lakes, we found that the most prominent shape of microplastic found is microfibers. So this is from a synthesis of, I believe, around 14 studies. So microfibers are thought to come from wastewater treatment plants, from people laundering their clothes, and then that moving from laundry machines in their effluent to the wastewater treatment plant, where much of it removed, but the volume of microfibers that move through wastewater treatment plants is so large that still some gets through. So fibers was the number one shape that's been reported. And then second to that was fragments. Those are a little bit harder to source apportionment because they can come from many things. But of course, one of them is common items that we use every day, food containers, takeout containers that enter the environment through the littering process.
Thank you, Eden. Karen, I'm wondering if you might provide a perspective, and others can can add around the human health dimension of the microplastics issue. Is there research being done to determine concentrations or presence of microplastics in humans? And what are some of the other dimensions of microplastics as it relates to human health that you'd know?
There is definitely global research being done on human exposure to microplastics and whether they're being taken up into our bodies, and a lot of work trying to understand the implications of microplastics exposure in humans. When we originally started this project, we talked about including human exposure, human health risks, and then realized that the science within the Great Lakes Basin was really limited. I mean, it's limited as it is for the ecosystem, but there was much less known about human exposures in the Great Lakes Basin. So we really restricted the scope of this report to the presence of microplastics in ecosystems and the risks to aquatic species. And what we did in our report is we made a recommendation to the IJC that they strike a work group next, sort of a phase 2 from our phase 1, that focuses much more specifically on what humans are getting exposed to, either from fish consumption or from drinking water. So we hope that'll be the next step from this first phase.
Thank you, Karen. Just an observation, there's a number of questions being posed that are excellent questions. They relate more to the actions of either or both federal governments as it relates to microplastics. The International Joint Commission is an independent advisor to governments. We are not part of both governments, and so there's a number of questions that are coming up that ought best be directed to the governments themselves. And some examples of questions I'm seeing: would the federal government, if Canada, consider adding plastics to the remedial action plans, the 43 pollution hotspots around the lakes? Are there opportunities for public input in the chemical of mutual concern evaluation process? So I'm wondering if, Rachel, you could drop a couple email addresses in the chat for folks, or otherwise make those email addresses available so folks can follow up on those questions, which are best directed to the governments themselves. There's a number of questions coming up around treatment approaches, and a couple of examples. The efficacy of wastewater treatment plants to remove or treat microplastics. As one example, another example is retrofitting washing machines or dryers to capture microplastics. I'm wondering if any of you might like to comment on sort of treatment approaches that hold particular promise that would serve as a complement to source reduction. Once they're in the system, how do we get them out of the system? Would anyone like to offer their perspective?
Start, if that's okay. We've tested quite a few of them. So in our research program, we've tested the washing machine filters, and they remove about 80% or upwards of the microplastics that are in our washing machines. Of course, only down to a certain size range that we measured, but they're quite effective. But note that you then you cannot take that material and put it down the drain. You absolutely have to clean it out and put it in to garbage cans. But we have suggested, not IJC, that governments consider that as policy moving forward, that you could certainly retrofit, or not retrofit, but you can buy the aftermarket filters or require that companies do it. I saw the question about wastewater treatment plants. There's been quite a lot of studies looking at how many microplastics are in wastewater at different levels of treatment. So wastewater certainly does treat microplastics. A lot of that material goes into the sludge. Then about 5% or less of it goes out with treated effluent into the environment. But of course, that sludge is sometimes land applied, so that's something to consider. My favorite tested solution are rain gardens or bioretention cells or permeable pavement, all those different types of treatments for urban runoff. They work for microplastics. They should be considered. The questions are more now around like, what is the cost to a city to upgrade to basically treat stormwater, and how often do you have to change them? And then Eden did a big study looking at microplastic capture by just simply using trash traps in at this point at industry for those plastic pellets being lost, and they certainly work. And then of course trash traps can also be applied in cities. So there is a lot of treatment. Of course, we want to prevent and ideally hope that plastics don't make it into these systems, but for microplastics that wear and tear off our products, treatment might be the best option. And so great question. And I do think it's something governments should be considering.
I'm going to hop on there and just say Eden mentioned the predominance of fibers, which come from wastewater, but Chelsea mentioned things like permeable pavements, rain gardens. Another major form of microplastics in the Great Lakes is tire wear rubber particles. And a third is foam from construction activities. And so different types of source control are needed for different sources of microplastics.
Thank you. Rebecca, while you got the floor, I'm wondering if you might comment on, there's a number of questions, and thank you everyone for your questions. To be honest with you, I'm having trouble keeping up, and I know others are as well. So no source of great questions. There's a number of questions related to the variability of microplastic concentrations across the basin. Questions and observations around the highest concentrations are Lake Michigan and Lake Ontario. Surprise at the concentrations in Lake Superior. Rebecca, can you comment on a couple things? One is, are those concentrations in any way related to the intensity of monitoring efforts? Is monitoring effort partly explaining the differences in observed concentrations, and also any perspective on how the concentrations that we see in the open waters of the Great Lakes might compare to other environments, for example, oceans, tributaries to the Great Lakes? So a couple questions there, but related to variability of concentrations.
Sure. So to start, I think bringing it back to our recommendations, that's why our #1 recommendation from the report is that we need, harmonized monitoring methods, and we need systematic monitoring across the basin. So there is great unevenness in the level of sampling effort that's been carried out in the different, not just in the different lakes, but also in the different habitats within the lakes. What's the tributaries versus the open water versus the sediment? And so when the sampling effort level is low, we have a lot of uncertainty around the values we're reporting. And so I would say in the lakes that have been less sampled, like Superior, I wouldn't say that we have a lot of confidence in what the environmental concentrations are. Michigan was one of the best sampled lakes, and so that was a lake where we'd have greater confidence. And Lake Ontario was sort of somewhere in the middle. And so finding the higher concentrations in Lake Michigan, I think we'd have high confidence in that. And in Lake Ontario, more monitoring might help us better understand like the spatial and temporal distribution of microplastics. But in lakes like Superior, where the sampling effort has been really low comparatively, I think we have much less of our confidence in those concentrations. And then as far as how concentrations in the Great Lakes compare to other ecosystems, I know we found that some of the highest reported concentrations in fish tissue come from Great Lakes. Maybe Chelsea wants to speak about that.
Well, I don't necessarily have to add anything of statistical value, but just from my experience, I started out my research in the middle of the ocean in the garbage patches, and then I worked in California on the coast. And when I moved here to Toronto and started working in the Great Lakes, I was shocked by the amount that we see in the water, as well as the amount we were seeing in the fish. So when I was doing work in the middle of the ocean, we found microplastics in 1 in 10 fish. And when I started working in California, it was 1 in 4 fish. And then when I came here, it was every fish. And it wasn't just a few particles anymore. It was like sometimes hundreds in an individual fish. So yes, I think by nature, being surrounded by big cities and being less dilute than the ocean, our concentrations are relatively high compared to those open ocean samples. Now in the San Francisco Bay, we also see a lot. That's a more enclosed area. So yeah, that's my experiential words of wisdom.
Thank you. There's a number of questions related to nanoplastics, which are of smaller particle size than microplastics. Would any of you like to comment on nanoplastics a little further?
Everyone's quiet.
Yeah, we intentionally, because there wasn't as much information about nanoplastics, we intentionally excluded them from the scope of our report, and were conscious when we defined microplastics for the purpose of the report. And so I think there's more emerging science on the effects of microplastics, but they're a lot harder to work with being smaller. One thing I would say is that, you know, our macroplastics, the large pieces of litter, break down to produce microplastics, and microplastics break down to produce smaller and smaller microplastics, eventually producing nanoplastics. So this is the problem with this persistent contaminant.
And I think I'll just add too, thanks, Rebecca, that the techniques to measure nanoplastics are really in infantile stage, I guess. They're under development because they're much harder to see with our traditional microscope or chemical analyses techniques. So I think we'll see much more in the literature on nanoplastics and, you know, where they are and what effects they're having, I expect in the next 5-10 years.
That's a really great point around the techniques for like lab analysis as well, right, Karen? So maybe it's a good opportunity to mention that there are pretty good standardized methods for enumerating microplastics and doing QA/QC, quality control among labs, but those aren't present for nanoplastics yet.
No.
Thank you. Eden, there's a number of questions about analytical techniques. And can you talk a little bit about how analytical techniques have evolved and improved over the last 10 or 15 years, and the degree of standardization of analytical / lab techniques? Any perspective on that?
Sure. So if you look back in a lot of the earlier microplastics literature, there wasn't much validation to determine if the particle that we were looking at was actually plastic. So I think that's one big advancement in the field is now it's standard to actually chemically identify your particle and confirm that it's plastic and not a natural particle that you are miscounting as a plastic. So I think that's one big advancement is this addition of chemical identification of the polymer that you're actually looking at. And then the second, I think that's really relevant to this risk assessment piece that we've been discussing is our ability to go smaller and smaller in terms of the size of particle that we're looking at. This, of course, is really important in the context of risk assessment because it's those smaller particles that are really available for the organisms in the ecosystem, and especially become relevant the size of the particle when we're talking about translocation. So Chelsea, correct me if I'm wrong, but right now I think with some of the lab protocols that Rebecca was mentioning, they can get down with confidence to, is it around 20 microns? With 20 microns, yeah. So there's still a gap there between, you know, the 1 micron, the lower end of the size range that we talk about in our definition, but that's a vast improvement over, you know, the history of the field. So I think that capacity to continue to measure these smaller and smaller particles, eventually get into the nano stuff, is a really impressive move forward, and tied to this idea of chemically identifying the particles, actually confirming they're plastic, is really, really important in the context of risk assessment.
I'll just jump in and mention that all of the SOPs we talked about in our webinar are in an appendix of our report. So they're in Appendix C. I know there's a question about, you know, are those methods available? And yes, they are. And then within the literature review, we reviewed all of the methods that have been used to collect microplastics in the Great Lakes, the field methods, the lab methods, and there's detail in there as well that talks about the quality assurance in the lab. And there's been a number of really excellent publications that describe how to make sure the data, the microplastics data, are high quality. So I would refer you to those documents because I think you'll find a lot of the information you're looking for there.
Yeah, we should say those lab SOPs exist and were produced by SQUIRP, who we worked with closely throughout this project. And then even some of our monitoring standard operating procedures build on the monitoring SOPs that SQUIRP developed, but tailoring them to the Great Lakes with different addenda. And so there's been like great collaboration with SQUIRP to try to develop these harmonized monitoring protocols, not just for the Great Lakes, but really like coast to coast.
I see there's a question about the open source data. So the environmental data we presented is publicly available on the University of Toronto's Dataverse, and there's a QR code in the slides that will take you to that database. And then the TOMEX 2.0 database will be publicly available, we hope, in the next several months.
Yeah, thanks, Karen, for that. And I was also going to observe that there's a lot of questions, very specific questions in the Q&A that there's related information in the project reports, which was a very voluminous set of reports that were produced, and they're all available on the Science Advisory Board website. Question about participatory science. Do you feel there's a role for participatory science in monitoring efforts, and how might the standard operating procedures that were developed be utilized or even modified and then utilized in that context? Any perspectives from the team?
I think that this actually exact question also came up as part of this work group, and I think that there is a desire for a lot of participatory or community science. And I think for microplastic sampling, it can certainly be done by groups if they follow the protocols. I think once you get into the lab, some of the equipment gets a little more pricey, and some of it's, like hundreds of thousands of dollars worth of analytical equipment. But I do think that when it comes to monitoring for plastic pollution, I do think that the macroplastic cleanups or trash trapping or any of that type of data is really useful for us to understand source and do source apportionment to inform upstream solutions. And I also think that the human eye can do really well down to like 1/2 millimeter. And so I think that there's a lot that we can do in community science, but I do not think it should be in the place of a monitoring strategy for microplastic, but I think it should 100% be complementary and included.
If I may, related to that, Chelsea, there were some questions related to sort of the relative level of effort and the resources that would be needed to adopt a basin-wide monitoring program and the associated analytical activity, the lab activity. Do you or others have a perspective on how that might be accomplished through coordinating and aligning with existing efforts and sort of leveraging all of the great work that is being done relative to Great Lakes water quality currently?
I think that I could answer this question, but I think Rebecca can do a better job at answering this question because I think she has a better idea of some of the monitoring work that already happens on the Great Lakes. So I'm going to punt if that's okay.
Sure. Yeah. I mean, I think the reason we're making the recommendation that Karen described about listing microplastics as a chemical of mutual concern and as a toxic chemical sub-indicator is that that would help insert microplastics into ongoing monitoring efforts. When we held our our first workshop on the monitoring SOP development, we had great participation from different NGOs, government agencies, and I think there was a really clear message that monitoring dollars are scarce. And so it's really important that we be efficient. And so we're not recommending that there be a new monitoring program that's purely focused on microplastics. We see that it's really important that it be integrated with the ongoing monitoring activities. And that's why we feel like the risk assessment framework needs to feed into the SOGAL reporting and why listing microplastics would be so valuable.
Thanks, Rebecca. A number of comments are coming in about you've been very clear that the standard operating procedures are for sampling. Chelsea, others, can you comment on standard approaches, standardization for the analytical or laboratory side of things?
Yeah, I can talk about what has happened with the SCORP work group. So the reason we didn't dive into the analytical in this IJC work group is because SCORP had done so much of it, and we didn't feel that we necessarily needed to reinvent the wheel. But as part of the SCORP work group, we did a really large round robin where we created samples that were clean water sediment samples, biota samples, and what we call dirty water, which would be like a manta sample from the environment. We spiked them with a known amount of microplastics that went down to 1 micron in size, and we sent them to labs around the world. We created standard operating procedures and asked them to follow them, and then we got back the data and looked at the accuracy and the precision among labs, as well as the blank contamination. That whole process went into creating standardized methods that California is in the process of. They've completed the process for clean water or drinking water, and they're in the process of standardizing for sediment, dirty water, and biota. And the idea is that those methods can certainly be adopted here. So those go down to 20 micron, which, as we've said, I think we didn't create methods with confidence below that, but a lot of our standard operating procedures that you'll see if you go into our work group report here really recommend almost just 50 micron because we think that that is the most feasible, that any lab could kind of turn on those methods now. So I know that other standardizing agencies are interested in different methods, but for now, I think the standard methods that exist are those that I mentioned.
And
oh, sorry, Karen, do you want to build on that?
I think the question too was about where to find those SOPs for the lab methods. And I think in our literature review, which is Appendix A, we reference a couple of papers that provide the SOPs for lab methods.
Yeah.
Thank you. Rebecca, did you have anything to add there?
Well, I was just going to say one thing we maybe didn't make clear about the importance of that minimum particle size that's being monitored and enumerated in the first, that's being sampled in the monitoring protocols and then enumerated in the lab protocols, is that with microplastics, we find that the smaller you go, the more particles you find. And so if you're comparing between two studies and one stopped at 1 millimeter and another went down to 50 microns, the study that went down, or the monitoring samples that went down to a much smaller particle size, will always find many more particles. And so one of the challenges in assessing the monitoring data is standardizing and kind of adjusting for that. And also that was an important feature in our risk assessment protocol, standardizing across the different ecotoxicology literature.
Thank you. And I'm mindful of the time. Perhaps one more quick question, and Karen, if I might direct this to you, and others can chime in as you wish. Number of questions around. It's been very well expressed that microplastics are a concern in terms of false satiation, in terms of direct toxicity, but the number of questions around microplastics sorbing other contaminants and being a vector for other contaminants into organisms, etc. Karen, any comments on that as we wrap up?
I'm going to pass that over to Chelsea, if I may, because she's done more work on this than I have. So Chelsea, please.
Sure. Yeah, I would say that there was a body of literature over the years that was looking at how microplastic accumulate chemicals. And we know that plastics accumulate chemicals. In fact, plastics are used often to make passive samplers to quantify chemicals in the water. So they absolutely will absorb, adsorb the different chemicals that are there. Then there's a question of, does that then make them more toxic? And I think that part of the reason why it gets a little fuzzy here is that a lot of natural particulate also accumulate those same chemicals. And so are plastics any more toxic than those natural materials when they accumulate those chemicals? Probably not in a contaminated system. I think what is really interesting when it comes to chemicals are the plastic additives and that are added to plastic materials to make them durable, to give them color, to make them flame retardant, etc. Those chemicals can certainly make plastics more toxic or be relevant to the mechanism of toxicity. But I think the complexity there is that there are tens of thousands of chemicals that are used in plastics in addition to tens of different types of polymers. And you very quickly end up with having to try to do this like very large battery of toxicity tests to understand. And so this is one of the reasons why we sort of put all the tests into one species sensitivity distribution and hope that it captures that variability. But I always have this burning question in my head of, is it enough? Is it possible that if I just made a toxicity, a species sensitivity distribution with toxicity tests from microplastic paint, for example, which has a lot of chemicals, would that shift the curve? So it's an excellent question. I think we're still trying to understand it, and hopefully that answered the question enough and gave a little bit of the nuance. But I think a lot of people are working on it, and there's still more to be done.
Thank you, Chelsea. And with that, I guess we'll conclude acknowledging the uncertainty and perhaps the hope of continuing our progress into the future. And so we have come to the end of our time, and unfortunately, we won't be able to field additional questions. So we'll wrap up now. Thank you to our presenters today, Dr. Rooney, Dr. Kidd, Dr. Rothman, and the future Dr. Hatley. Thank you to all our work group members, workshop participants, our contractor team, and my fellow IJC staff for their contributions to this report. To everyone who joined us on today's webinar, thank you for your time and interest in the work of the IJC and its Science Advisory Board. Further questions and information can be directed to the email shown on the screen, and a video recording of this webinar will be uploaded to the IJC Science Advisory Board webpage in the coming days. You can stay connected with the IJC on our social media channels. We also encourage you to subscribe to the IJC's quarterly newsletter, Shared Waters, to stay in the loop about IJC's ongoing work. Thank you for attending today. The webinar is now ending. Goodbye.